Okay. Thank you, Susan, and Paige, thank you very much for fostering with the NCI behavioral group my previous work and this work. Okay, let me jump straight forward. Before I start to describe our clinical study, I want to go back a little bit to show you the rationale and few points as introduction to the clinical study that we did. I want to make two issues as a background. The first one is that we believe that the perioperative period, although it is short, it is critical in determining long-term cancer outcome. And there is a period where there is a progressing tumor, but we are not aware of, and then there is a perioperative period where the tumor is being discovered and excised most commonly, and then there is a postoperative period where we hope that metastatic disease will not progress and will not occur. Now, we believe that the chances for progression of pre-existing micrometastases or new metastasis disease is increasing during this critical perioperative period because of many biological reasons that I'm not going to go into now, but also because of surgical stress responses and because of pro-metastatic, pro-inflammatory responses, both together cause immune suppression and impact the tumor microenvironment and directly impact the tumor tissue itself, as you have heard in many of the presentations. So all these things occur simultaneously together during this perioperative period, making it a critical period, high-risk period on one hand, but also an opportunity to eradicate or control residual malignant disease. And we recently reviewed these ideas, claiming that one should exploit this perioperative period to improve long-term cancer outcome. So that's the first point. The second point that I want to make is that we believe that both catecholamine and prostaglandins synergistically have a key role in this perioperative period in promoting cancer metastases. And to demonstrate this, I'll just show you very few of our previous animal studies. The first one that we used each of these drugs alone and together, we show that surgery indeed increased susceptibility to metastases. Each of the drugs alone reduced it, and together it was an additive effect. Since 2005, we were starting to think about a clinical study, and the first thing that we did is we changed and moved into a drug that we can use clinically, propranolol or etodolac. We choose these drugs for their general wide use and safety profile, and we found the exact same pattern of effect. Each drug alone was effective, and together more effective. Now, this is a very simplistic experimental metastasis model, and therefore the next stage was to move into a more relatively, a bit more clinically and biologically significant model. And in this one, which is actually probably our best animal study, we used a tumor which is implanted autotopically and metastasized spontaneously to different organs of the body. And we excised the primary tumor with surgery as in cancer patients. And we only treated the mice once during the day of excision with a combination of the two drugs or with each drug alone. And what we found is that neither of the drug alone was efficient in reducing metastases and in improving survival rates, but the combination of the drugs was effective. And we replicated this study with the many methods. We needed 3 times, twice with this model and another time with a different model with 3LL. The bottom line was in these more complex, more clinically relevant model, only the combination of the drugs were effective in improving overall survival rate. Two more model. One is liver metastasis of colorectal, and again, only the combination of the drug was efficient in reducing metastases. This was published this year. And an ongoing work in collaboration with Erica, we used yet a different tumor model, a human line that is implanted in nude mice, where we see primary tumor developed and metastasize spontaneously Once we see minimal metastases, the primary tumor is excised, and we follow only the metastatic progression in these mice. And in addition to this wonderful model that Erica introduced us to, we added at this period laparotomy, surgery, and the drug intervention. And what we found is that if you just remove the tumor without an extended surgery, then tumor regress and does not develop established metastasis. However, if you do laparotomy, if you add the large surgery, you have metastatic growth. And if you do laparotomy, but you also use the combination of the two drugs, you reduce the impact of laparotomy. So all this together gave us the notion that it is a combination of the drugs that we really need and want to use in human. As was quoted earlier, we also believe that all models are wrong, but some models are useful. And therefore, we thought it is really most critical to do the real study in cancer patients. And we are really thankful for the network for supporting us, for helping us to initiate this study. And I also want to thank Steve, which is not here, and Anil Sood, and Susan, Biobehavioral group at NCI that really not only supported us financially, but also helped us establish some of the protocol and forward study in 3 different hospitals in the Tel Aviv area. Okay, so we recruited 38 patients. We took the first blood sample. We assigned them to we randomized them to the drug versus placebo control. They underwent surgery. We took a second blood sample before surgery, a third blood sample after surgery. We took the tumor itself, and a 4th blood sample. The drug treatment was propranolol and etodolac, and we initiated it 5 days before surgery. We continued during surgery at higher dose of propranolol, and then we went down back to lower doses of propranolol for 5 days after surgery. Notice that at the T1 blood sample and T4 blood sample, there is no drug treatment. Therefore, these 2 time points are kind of baseline reference points with respect to T2, T3, which have both drug treatment and surgery. Okay. Study outcomes. We looked at the 4 blood sample. We looked at the 4 blood sample. We analyzed serum level of soluble factors, cytokine, cortisol. We did FACS analysis to characterize leukocyte population, ex vivo induced production, and messenger RNA profiling with the aid of Steve. Looking at the primary tumor tissue, we analyzed whole genome RNA expression and immunohistochemistry to be conducted in the future. And we look at adverse effects. Of the 38 patients, 2 had what one would call non-serious, potentially drug-related adverse effect. 1 was placebo, 1 was control. The placebo had a greater level of adverse effect than the drug, and the drug safety profile seems okay based on this small number of patients. Okay, results. Corticosterone level, as would be expected, increased from T1 to T2, awaiting surgery. This is surgery, this is a treatment. No drug effect. IL-10 increased from T1, T2 to T3. So both the corticosterone and IL-10 increase in awaiting surgery under a condition which one has stress. But again, no drug effect. When we look at the pro-inflammatory responses, this is where it becomes more interesting. IL-6, one can see that there is the control group increase from T1 to T2, as one would expect with the stress. But the drug treatment prevent and reduce these increase, creating a significant and large differences. The same occur with CRP, another pro-inflammatory marker, and both CRP and IL-6 have prognostic value in breast cancer patients. Higher level predict higher mortality rate. If you one look at the more molecular level, intracellular level of peripheral PBMCs, one see NF-kappa B, for example, as one of the transcription factor which is being reduced by the drug treatment, as other transcription factors that are also drive IL-6 and other pro-inflammatory cytokines. So these findings are also echoed by these intracellular levels of molecular control of this pathway. Okay, here comes an exciting part, and Steve is gone, so but he saw these results many times. So, and I'm really very thankful for Steve for introducing us to these approaches and helping us conduct some of the studies and some of the analysis. We found more than 200 genes which are differentially expressed by the drug treatment, 200 increase, 200 decrease, more or less. Because one measure all messenger RNA, approximately 30,000 in the cell, one expects that some of these changes are false positive, okay? But some of them are real. The problem is that we don't know which one is real and which one is not. Therefore, one has to have an a priori hypothesis regarding what does it mean, all these changes. And what we did, and with the help of Steve, we defined three different levels of hypotheses. The first relates to the messenger RNA levels themselves, and we looked at a characteristic of the tumor, which is called EMT, epithelial to mesenchymal transition tumor cells that in the process of metastasis need to transform from epithelial characteristic status into a more mesenchymal structure status of tumor in order to extravasate the tumor, detach from the tumor, intravasate into the blood vessel, and extravasate into remote organ. Without this EMT, it is very unlikely for a tumor cell to metastasize. The EMT is now known as a critical factor in the metastatic process and as a predictor of later metastasis. So we looked at this characteristic in the tumor based on a priori hypothesis and based on knowing what the exact messenger RNA profile in EMT cancer cell. And we looked for this particular signature within the tumor. And because we are telling you, you know that what we found is indeed that the drug treatment reduced the mesenchymal status of tumor cells and did not increase, but at least remained the epithelial status. So overall, this is a good prognostic marker. We also looked at tumor infiltrating leukocyte, and there is a decrease in monocyte number and increase in B cell numbers, and both changes are known positive prognostic factors in breast cancer patients. Monocyte, we've heard a lot about being transformed to M2, and B cell are protective in the breast cancer tissue. And the last thing that we did is we look at a variety of transcription factors in the tumor, and this a priori hypothesis regarding transcription factor addressed characteristics of pro-inflammatory, pro-metastatic, and survival benefits, again, based on a priori knowledge and hypothesis. And we found many of them to be reduced by the drug treatment. This includes GATA1, 2, 3, the STAT3, the EGR, and GRE. Okay, so this is really very encouraging. Not all the outcomes that we have indicated, the effect of the drug. For example, if one goes back to the serum, you can see three pro-inflammatory markers, IL-8, IL-18, and TNF-alpha, which were not affected by the drug treatment, and their pattern of response was different from the IL-6 inflammatory response, which was affected by the treatment. We also measured several immune indices, and what we found is, for example, interferon gamma-induced production and IL-12-induced production in PBMCs are known to be reduced by stress and surgery, and they were indeed reduced here, but the drug treatment improved this status and had no effect on the anti-inflammatory IL-10 as we would like it to be. When we look at NK system, we look at CD11A, a molecule that helps assist NK cell identified here and kill tumor cell. And the drug treatment improved CD11A, increased its expression on NK cell in the periphery. And the reason that we did study this marker is because in the previous studies that I showed you, where the combination of the drug improved survival rates, we also measured CD11A, and we indeed find that in this model system too, the drug treatment improved CD11A expression. The last thing that we found is just the morning after surgery, there was a large increase in the classical pro-inflammatory monocyte in the blood, and the drug treatment reduced this response, had no effect on other type of monocytes. And we also conducted exploratory analysis on some transcription factor in PBMCs, which are known, transcription factors which are known to relate to tumor progression and to survival in breast cancer patients. All these are known to impact cancer progression where they are studied in the tumor cell. We studied them in PBMCs, and they were reduced as we would expect them to be. This one was also reduced in the tumor cells themselves. Okay, so what we believe is happening is like that. Even before surgery, there is psychological stress, which increase sympathetic activity, which increase pro-inflammatory responses, which increase transcription factor that have either inflammatory, pro-metastatic, or survival predictive ability, which then elevate messenger RNA level, change tumor characteristics such as ENT, and potentially increase the metastatic potential of the tumor. All this occur even before surgery, just awaiting surgery, being stressed before the day of surgery. Simultaneously, and just after that, we have also the surgical stress and the perioperative psychological stress, which together increase both prostaglandin and catecholamine, which cause immune suppression, impact tumor microenvironment, and directly impact not only the tumor cell, but also the residual disease that remain in the body after we excise the primary tumor. And in this study, we showed all these processes. We showed increased IL-6, we showed increased transcription factor, etc. We do not know what happened with respect to long-term cancer outcome, to metastasis. When we used the beta blocker, we showed that the beta blocker and the COX inhibitor, the combination, reduced all these processes, but we still do not know the most important issue of whether this treatment will also improve long-term survival. So this remains to be seen. Summary. The perioperative period is one critical period that one needs to approach and to treat patients during. One can use different approaches during the perioperative period. We used the blockade of beta-adrenergic and COX inhibition in order to reduce their impact on immunity on the tumor. Such intervention could transform the perioperative time frame from being a prominent facilitator of metastatic progression to an opportunity for arresting and or eliminating residual disease. And the most important, large-scale clinical studies are needed to assess long-term cancer outcome in various types of cancer. What is next? We hope to continue this work and to conduct larger clinical studies with long-term cancer outcome, hopefully in breast, pancreatic, and colorectal cancer patients. And we also want to incorporate behavioral intervention and psychological intervention during the perioperative period, because the majority of the patients are excluded from the study given medical consideration. Because we believe that the combination of perioperative psychological behavioral intervention and pharmacological intervention might be more efficient than any of these approaches alone. So I just want to thank my students, and again, the group and the Israel Science Foundation that also supported this study. Thank you very much.